Okay, guys, as promised, here is again the quick reintroduction to chapter two. I know we haven't met in quite some time, but I wanted to reintroduce chapter two, the overall objectives. And what I'm going to do is I'm going to probably break this up into two 20-minute segments of presentation. Hopefully, that should get us through about slide 30 so that when we meet on Monday, we can finalize them. So, unfortunately, that means, and I will post this as an announcement, I'm going to have to either move the blood spatter lab or we might have to drop the blood spatter lab altogether. The weather really threw us for a loop this semester. So, needless to say, to be continued on that. But with that being said, let's make sure we don't fall too far behind. So, chapter two, bodily fluids and waste products, right? So, again, our high level overview, we are chasing PMI, but now we're starting to transition into some of the biological um, foundations that we're going to use throughout each chapter for the methodologies to be able to get us back to that PMI. So we're going to distinguish, describe, and explain different aspects of blood cells and how we can detect blood. We're going to look at the composition of blood at crime scenes and potentially determine how best to quantify or identify the assailant versus victim. And we're going to look at saliva, semen, urine, and feces as a overall forensic indicator. So why this matters as far as chapter two, these are some of, again, just sort of the questions as you move through chapter two. Maybe this is something you want to come back and revisit, right? What are things that blood can and cannot tell you? What are presumptive versus confirmatory types of tests that we see at the crime scene versus those we have to take back to the lab? How best do we document and collect, and how do we best protect the interpretation of that as we move towards prosecution? We're going to look at different types of evidence. So we're going to look at different surfaces, stains, and the limits of a blood pattern analysis. We're going to look at, again, other fluids and waste products, and we're going to potentially try to use these as building a defensible narrative with the fact that everything has uncertainty, right? There is no 100% guarantees. There always is potential error. And again, think about what types of errors that could look like, or what do errors look like? Humans themselves are a big contributor to those errors, right? So the big mindset to keep in mind here is treat every test and every tool as part of this decision-making process. This is not an automatic truth. So again, I'm not going to spend too much time here. This might be where we end on Monday when we meet. But again, this was the scenario that was proposed to you all in your groups. You were starting to work on that. So I'm not going to spend too much time here. But again, we were going to lead with what would be the final, top risk that your group could come to based on the scenario that would lead to a false conclusion, right? That could give you the incorrect. Because just like in science, right? And the basis of this course is the foundations of biological principles, right? The study of life, how we know things to be living, the characteristics, the mechanisms that we know of life as the foundations for understanding crime scenes, right? Just like in the scientific process overall, a null hypothesis, something that disproves your hypothesis, you finding out your hypothesis is in fact incorrect, can be just as important. So, things to consider, right, as we're moving through. And again, we'll come back to this as a group on Monday. Okay, so now we're going to just take a step back. We're going to do a little anatomy and physiology. We're going to do a little biology here. We're going to talk about cell characteristics of blood. We're going to look at the main aspects of blood and what defines blood and what blood is made up of. So, what do we know blood to be responsible for? Blood is responsible for transportation of gases, mainly oxygen and carbon dioxide. It's not the only ones, but it's mainly those two. The transportation and removal of waste products, processing by liver and excretion out, right? Removing anything that we need. Um, Keeping the good stuff in the body, water retention, fluidity, osmosis, plasma concentration levels, pH, all of those things. Transporting of nutrients, and again, the removal of toxins overall from the body. So if we are thinking about this as far as crime scene makeup, and also just, what do we have to understand about the makeup of blood? We have to understand what is the breakdown of what blood is physically made up of, right? So first and foremost, blood is a tissue, right? It is a circulating tissue. It's important. It's a tissue that continuously moves throughout the body, and it makes up about 8 to 10% of the total body weight. Normal conditions, again, remember biological swing, environmental conditions, et cetera, disease states, gender, not gender, sex, I apologize, age, right? All of those things. But normal adult blood volume is around 5 liters total. Females roughly between 4 to 5. Males, because they tend to be bigger and have a larger muscle mass, 5 to 6 liters. And it is made up of a multitude of cellular material in a suspended fluid known as plasma. And again, we can break this down even further, right? We can take a test tube of blood and we can centrifuge that blood, spin it down at a very high rate, and we can separate it into the different concentrations. What we'll see is majority outside of the makeup of the fluid of which the blood cells are suspended in, right? Plasma being the biggest concentration by volume, is that we see that mainly we are made up of red blood cells. And again, our red blood cells are incredibly important. We need them in a high concentration. Why? Because they carry oxygen for us, right? And then right here, what do we see? That this very thin band, right? Looking at, breakdown there, very low percentage. But these are all of the things that are also incredibly important, like our white blood cells, platelets for clotting factors, right? And we can break them down even further. Well, what I want you to know, I want you to know the general makeup of blood. I would want you to know that plasma, this suspended liquid, is the majority of what the liquid component of blood is. That red blood cells are the largest concentration of cellular material. And then we have these other formed elements of the white blood cells and platelets. And then, of course, this information down here, right? The general makeup between males versus females. That's important if we're looking at, blood loss at a crime scene. And overall, just makeup when we're thinking about, if we come to a scene, sometimes, accidents happen, things take place. If we're trying to determine the timeline of things, maybe blood loss leading was the cause of death, right? So again, knowing the general makeup between males and females. So let's do a little bit of a deeper dive into the blood cells themselves. So as I said, the red blood cells, as far as cellular material, are the main makeup of that blood, right? Of the cellular material, I should say. We can also call red blood cells erythrocytes. That is their official anatomical name, physiological name. And their function is, of course, to carry oxygen. They are the main, they are the cell that carries and binds to oxygen and removal of waste product of carbon dioxide. This is very important. Contains a nucleus. No, it does not, which means what? I don't have DNA, right? I'm not containing nuclear DNA here. I also do not contain a mitochondria, which is very unique. Very unique cell as far as the body, because mitochondria, as we'll come to find out in chapter 3, is another place we can turn to for DNA, right? So these are cells that are mainly just holders of oxygen. Then we have our white blood cells. This is, again, part of the other elements of cellular makeup. These we can call leukocytes. Leukocytes are broken up, as we saw in the previous slides, right? If we are looking at our white blood cells, we can look at lots of different types of white blood cells. These are all our immune defenders, right? So again, white blood cells mainly as an umbrella term are immune defenders. And absolutely, they have both a nucleus and a mitochondria, right? Platelets, we can also call these thrombocytes. They are very vital in the clotting factor, right? So if you are on blood thinners, right, an individual may be on a blood thinner. What would that lead to? Higher blood loss, right? This is why elderly patients, if they are on a blood thinner for whatever drug they may be on, recent surgery, a whole host of different things, right? They don't clot very easily. They have to be extremely careful. The smallest bump, bruise, nick can lead to very serious bleeding. And again, where would I be going to look for DNA? Well, in platelets, I'm not going to have a nucleus, but I do have a mitochondria. So again, out of this slide, right, a question would be, which of the following is not a good place or a good cell to gather DNA evidence from? It would be the red blood cell, right? Because they do not contain a nucleus, nor do they contain a mitochondria, right? True, false. White blood cells, I can go to both the nucleus and the mitochondria for DNA. True, right? True, false. I can go to platelets for both nuclear and mitochondrial DNA. False, right? And so this is also important too, because as we understand blood, and even with things like our white blood cells and immune response, we may be having a reaction to something. Were you given something that is a known allergen to you? Was your immune system leading you into things like anaphylactic shock, right? So the immune system is also another really important place as far as our white blood cells. Again, to understand potentially what was taking place with inside of our victim leading up to death, right? So the immune system itself is responsible for being introduced to any material, foreign or inside, that can provoke the immune response. So mainly this is going to be something that is a foreign defense system, right? Something that's being introduced. Again, we're starting to understand more about cancer and turning on the immune response as well, which is really important. It is attached to the surface of, it attaches to the surface of red blood cells, and it kind of hitches right through. And when we think about really what we're talking about as far as our immune system, we're talking about things like antigens and antibodies. So antigens are anything, a protein, a carb, a lipid, that is a name tag that can produce, an immune response. So anything that can trigger the immune response to turn on. The antibody is what your body sends out. It recognizes. It says, oh, I've seen you before, and it wages war against the antigen, right? So the antigen is introduced, and if your body has not had exposure to this before, it's going to take a little bit of that antigen in, and it's going to, right, it's going to first bind to it. If we've never seen it before, the immune response may not have the appropriate antibodies to fight against it. So what is it going to do? It's going to, the body's going to need to bump into some of these immune cells, and the immune cells are going to take little pieces of this foreign invader, and they're going to try and take it in and hold it as a memory, quote unquote, inside of the DNA, right? Because again, this is our immune system response. It has both the nuclear DNA and the mitochondrial DNA, if necessary, to be responding. right? But mainly, where are we storing it? We're going to store the exposure with inside the nucleus of the white blood cells, anything that's involved in our pathway of fighting defense. And so what does that mean? It means that over time, if I am exposed to that antigen again, right, my immune response is going to bump into these. It's going to say, ah, yes, I recognize you. And it's going to start sending out the appropriate antibodies to bind to this antigen and dismantle it so that it cannot bind to your immune cells and cause an immune response. And so what do we see? We see that when we are born, we have kind of an innate immunity built in. You have these things that are a part of your bloodstream, right? And then over time, and these are really part of your rapid response. But as more time is exposed and you are exposed to new things and you're building your immune cells and you're building up your immunity and you're building yourself up different antibodies to different antigens as you're growing, right? Over time, your adaptive immunity starts to build up further and you start to put things into the memory bank, right, of that DNA. So that over time, you can eventually say, I've been exposed to this before, and I'm going to navigate this through the release of either antibodies, or if you are looking at things like long-term viruses or even cancers, you are going to move into these secondary and tertiary cells that are really, really going to be ramped up as part of your large immune response. And again, all of this is important because I can test for these to see potential underlying states of an individual, right? I can do this within, pathology, toxicology. I can look to see what was going on in this individual. Again, if I have these tissues available at the scene. So this leads to then very important work that became Dr. Carl Landensteiner's work, and he was determining the blood system, the ABO system in 1901. And again, it was looking at the antigen antibody system carried by red blood cells to recognize foreign invaders, or most importantly, types of blood, right? Because your blood cells themselves carry specific antigens antigens, I'm sorry, that are going to help in response to immunity, response to clotting factors, etc. So what do I mean by that? If you have type A blood or type B blood, your red blood cells are going to carry an agglutinogen, right? An agglutinogen antigen to that particular blood type, right? So agglutinogen antigens on the surface of the blood cells, these are those that stimulate the production of the agglutinin antibodies, which triggers the immune reaction. These are important for determining your blood type, right? And I've got a picture to come in the next slide. So if I carry A or I have type A, I can only carry and mark for recognizing type A, right? If I have type B, I can only recognize and play well with type B. If I have type O, I carry neither A nor B. But if I have type AB, I carry both A and B. So let's look at why this is important, right? So for type A, my antigen carrying marker is for the antigen of A, right? So if in the antibody, I am anti-B, I cannot bind to anything with B. I'm going to fight off B. I create antibodies against B, right? Such that if I was introduced to A, these would go, or if I was introduced to B, these would bind to and lead to serious clotting factors, and my blood would no longer flow freely throughout. Same thing if I had B and was introduced to A. I'm going to distribute antibodies against A, right? So what does that mean? I cannot have B or AB blood, because even though there's type A there, it still carries type B with it. So again, I can't have that. I can have type A blood or type O, because again, what does type O have? It has neither antigen of A and B, so it's easily accepted by A or B, right? What about antigens A and B? Well, I don't technically carry an antibody to either. So I can have A, I can have B, or I can have AB. I am the universal recipient. Nothing that my blood makes an antibody against it because it recognizes A and it recognizes B, and O carries neither A nor B, so I can have O too, right? But what about O blood? O blood carries the antibodies against both A and B. So I can only ever receive O blood. Therefore, I am the universal donor. And again, that would be a great question. Which of these is the universal donor? Which of these is the universal recipient? Why can't type A have type B blood? Because you have antibodies against B blood. Or why can't type B have A blood? It has antibodies that will bind to and clot up that blood and not allowing for it to work appropriately, right? So if I clot up the blood isn't flowing, it can lead to, stroke. It can lead to many different types of blood conditions and clotting factors. It can lead to a large clot, right? It can lead to clotting to the lungs, is a very detrimental thing. So why is this all important? It's important because again, we can also look at the variations of blood typing with inside of different races. So certain races tend to have higher proportions of blood groups. Again, this just makes sense by genetic composition, genetic isolation, and selection factors of mating, right? Um, so again, we can look at these things. It's not an end all be all. Remember, we're just collecting the information. We're going to use the information at hand, and we're going to again relate that back to how much information do we have from the deceased? How much information does the scene tell us, right? I might not be coming up to a scene I might be coming up to a scene that's 50 years old, a cold case, where all I have now is dried blood, where I have very old blood. And I might not even be able to know what the particular race or, makeup of this individual is. Again, these are all tools that I can have to my benefit. So as far as breakdown, we know that AB is more common amongst Japanese individuals than it is for Europeans. Europeans carry AB at about a 4% rate, and Japanese individuals at about a 10% rate. Now, those aren't extremely high numbers, but there's something to start with, right? We wouldn't use this as the foundation of everything when we go into the courtroom, but this is something that we can use as starting metrics, again, especially for things like cold cases or blood at a scene that is not the victim's. We also know that there are blood disorders that are unique to certain races and populations, right? Sickle cell tends to be almost exclusively found in individuals of African descent. And again, what does sickle cell do? It creates a sort of unique sickle, right? If you think of kind of the Grim Reaper, I know I'm not in class right now and my face isn't being shown, but if you think of the sickle, that the Grim Reaper is always kind of shown with, it has that sort of shape, hence the name sickle. So these they can be very dangerous. Sickle cell can be very dangerous because it doesn't carry as much oxygen as individuals that have normal spherical donut shape cells. But there is actually a benefit to this. And what they have found is that sickle cell is a protection against the plasmodium parasite that causes malaria. And where is malaria very prevalent? In African countries. So again, this has served a purpose. But we can also say that again, if we are finding characteristics of blood that has a sickle cell shape, we are almost certainly dealing with someone who has an African descent of some sort. We also see that many antigens and enzymes in the blood exist in multiple forms. We call this polymorphic. Poly meaning many, morphic meaning many different varieties of, right? Your unique biology makes your blood profile extremely unique. So, again, I'm not going to expect you to know this per se. I'm showing you this just to, again, simply to state research has been done on this. Research has absolutely looked at, are there ways that we can use blood profiling, blood typing, as a way to understand the percentage within a population of that type of blood? That could help us to rule out certain things. We could look at percentage of US population broken down by different ethnicities, right? All of these things are merely just resources to helping build that case. Okay, I'll end with this one and then we'll pick up with the chemistry of blood and how we detect it in a minute. So, as we move into this next set of slides, again, we need to consider how best are we going to collect the blood? How best are we going to analyze this blood? What are we hoping to gather as information to say something about, again, building our case? So, questions to consider. And this would be kind of maybe an essay style or short answers type of question. What are some considerations detectives or forensics and investigators need to consider when they're analyzing blood at a crime scene? We need to consider do we have enough sample? Is there enough for us to physically collect? And is there enough available that isn't fully degraded? Blood is at one point had living cells as a part of it. Now, again, our red blood cells are a little bit more of an advantage there, but still, the environment can break these things down. They have cell membranes, right? We need to know, are we looking at potentially victim or assailant? That will come, that'll be considered when we continue to move through looking at the different variations of pattern analysis. We can maybe back at the lab understand, is there a mixture of blood that we're seeing, right? Maybe I'm looking at a universal donor or a universal recipient, and I'm looking at that because I'm seeing some variety in the blood itself. That might be something I need to take into consideration to not identify the wrong person. Big one. You wouldn't believe how big. Animal versus human blood at a crime scene, right? Especially if there was an animal protecting a loved one. Sometimes as simple as even person was cooking steak or handling meat, right? And then of course we're looking at other potential fluids that could be at the crime scene. So the goal, screen efficiently, document thoroughly, and again, make sure we are protecting the downstream possible outcome of DNA analysis and or toxicology from these reports. Okay, I will be picking up in the next set of slides to carry us home until we meet on Monday.